We are live once again with the Organic Consumers Association and me, Pat Thomas. Wherever you're joining us from today, thank you so much for tuning in. We're talking extreme science today, as if there wasn't quite enough crazy out there at the moment. US scientists have announced that they've created an organism that's part human, part mouse. This kind of organism is known as a chimera, and depending on your point of view, it could either be super cool science or science of a much darker and more disturbing kind. But beyond that, it's also the kind of extreme science that brings with it a whole host of ethical and safety considerations. So to talk about some of these with me is Stuart Newman, professor of cell biology and anatomy at New York Medical College at Valhalla. Stuart's also the author of the book Biotech Juggernaut, which in part details his own efforts to raise some of the ethical and safety considerations around chimeras within the scientific and regulatory community. Stuart, it's great to have you back.
Thank you. It's nice to be back.
Yeah. When you were last with us, we were talking about COVID-19 and the possibility that the virus was a chimera. Does the creation of a human mouse chimera have any touch points with the creation of chimeric viruses, or are they completely separate issues?
Well, it's a classic term, actually, out of Greek mythology, and it refers to organisms that are pieced together from parts of other organisms. So in the case of the virus, it was the genome of the virus that
Stuart, I think we've lost you there.
Yes, I'm here, and now you're back. Yeah, okay.
There you go.
Okay. So it seemed to reflect passage through several different species, and the genes came from viruses that originated in different species. In the case of the human mouse chimera, it's not a genetic chimera, it's an embryo chimera. So what happens is that embryo cells from humans and mice are mixed together, jumbled together, and implanted into a mother mouse and develop. And they could potentially, depending on whether the organism, the mother can accommodate them, they could potentially develop into full term. So they would be full-term organisms that are part human and part another species. It's been done with pigs. Now it's done with mice. And there are various twists and turns in the different species. But the purpose of it is to make experimental animals that possibly drugs and viruses could be tested on, or just to understand how development, how cells can communicate during development. And the chimera shows that they can communicate across wide species boundaries. I should point out that a chimera is not the same as a hybrid. So if two animals mate, like a horse and a donkey, for example, you can get a mule. That's a hybrid. And in a hybrid, every cell of the animal has characteristics of both species. So every cell of a mule has horse cells and donkey cells in it. In the case of a chimera, the organism that develops is a mosaic in the sense that each cell comes from one or the other species. So it really shows that cells can communicate even with their own genes across species lines.
It seems so far-fetched. Can you say a little bit about what the purpose of the study was? I mean, beyond just showing that you can do it, was there a purpose to the study? What did it show?
Yes. So these investigators are interested, among other things, in making animals that tissues and organs could be harvested from. So you can imagine if you could do it with a human and a pig, or a human and a monkey, that you could have free-living organisms that are part human and part animal. And it's been shown when this was originally done back in the 80s with sheep and goats, they generated an organism called a geep that you could actually graft tissues from the chimera into one or the other originating species. So imagine if you had an animal that was chimeric, and you could use that animal to graft hearts or graft livers into humans. That's one of the objectives. They also say in this new paper that they could make human lung cells in a mouse and test whether the coronavirus, for example, can infect and how it can infect. So you can create experimental animals that don't have the same strictures against them that you would have for experimenting on humans. They could experiment at will on these animals.
So, according to the report, the embryo was around 4% human cells, which the researchers claimed was the highest percentage to date. But post-publication, I noticed they suggested that the ratio could be even higher. Is there a line? Is there a line, a percentage of human cells that's okay and ethically okay and safe, and a percentage that shouts out danger, or are we still sort of feeling out the lines of this?
Well, I think that it really, the line is very fluid, and it all depends on what the public is ready to get used to. So, for example, in 1997, I was very concerned about this, and I applied for a US patent on a human, non-human chimera. At that time, there were no experimental human, non-human chimeras, but there was the sheep-goat chimera, the GEEP, and I used that as a prototype. And I applied for a patent and simply to raise the issue. I had no intention of making the chimera. And to apply for a patent, you don't actually have to do the thing that you're patenting. You just have to describe it very well. And the patent was rejected. They had to go back to the 1820s and find a standard that would allow them to reject the patent. In the 1820s, one of the patent commissioners said that patents can't offend public morality. And they said that my patent, if it were implemented, would offend public morality because, and this was the modern term, they said it embraced a human. Basically, there would be no way of restricting how many cells from the human would be in the chimera. And still there's no technical way to determine that. They're doing a lot of trial and error. There is an unpublished paper that's circulating now from an Israeli group that claims that they made embryos with 30% human cells. So you can imagine if it's 50% human, 70% human, is it human or is it not a human? Can you experiment on it? Can you not experiment on it? And for this reason, I actually have come to the conclusion that this work should not go on. I think it's too disquieting and too disruptive of basically what we consider being human is.
No. It's interesting because as you're talking there, it occurs to me that many of us believe that the biological boundaries between species are pretty firm. Like there's a Chinese wall between us and a mouse, for instance. But what this research would suggest is actually, as you say, they are quite fluid. Apart from the kind of yuck factor, I suppose, that a lot of people might feel, is there a kind of a touch point for disease, for instance? If we create these chimeras, does that also break down the barriers of disease in human beings, making them jump more easily from species to species?
It definitely would. If these animals were farmed, if they were kept, there would be new kinds of viruses that would develop that were characteristic of one species, but would readily mix very intimately with the other species. I can say something about the species barrier, because one of my other hats, the main thing I do is what's called evolutionary developmental biology. So this is a field that I work in. And the idea that there are rigid barriers between species that are kind of evolved and once established can't be crossed. That idea is really kind of dissolving now. People recognize that larger, broader groups like phyla, like we're part of the vertebrate phyla, or classes, we're part of the class of mammals. Those groups have a kind of a biological reality in the sense that they are truly different from one another. And they use different developmental means to generate their members. They evolved in an earlier time. Species are more fluid, and people are very familiar with the sterility of mules. So they say when species mate, that the result is sterile, so it doesn't go on. But in fact, that's an exception. Like in birds, species can mate and produce new species. So the species boundary is not a rigid boundary.
And this is a major ethical point, isn't it? As to, if we take this to its sort of extreme end, are these characteristics heritable? Can they be passed down from generation to generation?
Okay, so in the case of a hybrid, like the hybrid birds that I'm talking about, they can be. In the case of chimeras, they can't be, because if two chimeras mate, and they could, the geeps were mated with each other. And what do they produce? They produce one species, because a sperm of the goat meets up with the egg of a goat in the two chimeras, and they produce a goat. Or correspondingly with the sheep, they could produce a sheep. Or in the case of sheep and goats, the sperm of one species could meet up with the egg of another species, and they could form a hybrid. But in none of those cases is it passed down to further generations. The interesting thing about the sheep-goat experiment is what it showed is that chimeras do produce gametes of the originating species. So you could imagine, this is one of the most disquieting things about the new work, is that if you had mice that were chimeric and they produced sperm or egg of humans, which they could, you could have a colony of mice that you could mate and produce human embryos. So somebody could have a company where they've gotten movie stars' cells and turned them into stem cells and made mice that are producing sperm and eggs characteristic of those celebrities or movie stars or artists or whatever you want. And you could go and buy eggs or buy sperm for artificial insemination, in vitro fertilization, and basically have a company that makes designer gametes.
And so by sort of cutting out the human element of that, just sort of churning them out in a sort of a factory of animals, if you like. I suppose some scientists think that they are getting around the ethical dilemma of this, but I would suggest that it's creating even more of an ethical dilemma.
It's creating, I agree with you, it's creating a huge dilemma. I mean, basically, you would have factories that are producing human embryos, and there would be no basically egg harvesting involved. The eggs would be made in the mice, and the sperm would be made in the mice, and you could just specify who you want and get the gametes that you can afford. Also in these colonies, the mice could mate with each other and just produce human embryos themselves.
Mm. So you mentioned a little bit about the patent office response that they said no, but it wasn't really quite as straightforward as that, was it? There was a process, as you kept going back trying to test the boundaries, didn't you?
Right. It went on for about 7 or 8 years, and they originally said that it offends public morality, so it's not patentable subject matter. I kept on rewriting the application so as to kind of get around some of these objections. There were technical objections, and the technical objections were, could I predict ahead of time what proportion of the resulting animal would be human? And there's really no good way of predicting that. And certainly in 1997, there was no way of application eventually fell on the basis of a kind of technical specification. It needed to be specified more strictly. So they disallowed it. And there were about three or four different submissions. And then finally, I just decided not to reapply again. So the ultimate fate of it was to be rejected. But as it was rejected, it also made a lot of this technology not patentable anymore, because once something is publicly disclosed and in the public record, you can't patent it anymore. It's not novel. So there are some novelties in the new technology, like the paper that we're discussing has done a new twist. It turned out that even though you could make human pig chimeras pretty readily, and some groups are working on that, human mouse chimeras was more difficult because the mouse embryo cells were in a paused stage, and the human's embryo cells were not. And nobody knew quite why. But what they did is they used a drug to basically put the human cells in a paused stage. And then when they mixed them together, they cooperated very well together in a way that they didn't before. So they've overcome a big technical obstacle in making human mouse chimeras.
I'm wondering what, as you were doing this, as you were going through this 7-year process, what other scientists and other government officials thought about what you were doing. Were you cheered on? Were you criticized? What happened there?
Most people just didn't say anything about it, but there was a very public pronouncement. Apart from the patent commissioner who invoked this public morality thing, the chairman of the Harvard genetics department at the time was interviewed on radio, and he was quite outraged that I would even suggest that scientists would do this. He called it outlandish. He said that it's just scaremongering to think that anybody would make a human non-human chimera. And so this was years before anybody really tried it. But it was being done in other animals, and it stood to reason, given the press of commercialism and basically the incremental acceptance. In the book, my book with Tina Stevens, Biotech Juggernaut, we discuss this issue and a number of other issues, including human germline genetic engineering and several other biotechnology issues that relate to human biology, in which at first people were very reluctant to accept it. Then it was kind of touted and advertised, sometimes under false pretenses. Promises were made, and finally, in California, there was a big initiative to allow human embryonic stem cell research that the federal government wasn't allowing at the time, and multi-billion dollars was spent out of tax money to fund these labs. And eventually the labs took another turn. They started working on a different kind of stem cell that didn't involve human embryos. So the whole pretext of the California Institute of Regenerative Medicine was basically not implemented, but the money was spent.
And that brings us back to a familiar refrain, really. I mean, the science marches on. We find ways to get around species barriers or get around, barriers where there's a timing in the development of an egg or what have you. And public money is being pumped into this, but where is the public good? I ask this question a lot, but I still don't see it. Where is the public good for this? Public money for public good.
Yeah, I think the public good is not obvious. So take an example in the paper from the Dr. Feng's group in Buffalo that announces the mouse-human chimera. They talk about making mice that have human lung cells and testing viruses on that. Well, you could test human lung cells from explants, biopsies of human lung in a dish, and you could see whether the cells react to the virus. In the case of a mouse, if you did it, the mouse has its own immune system, which is different from a human immune system. So you're getting a very complicated assay system that's very different from the human one. But a lot of science is presented in a very reductionist way. Oh, if you could put human lung cells in a mouse, then you have a model for studying human lung cells. Well, it's not so simple. But the thing is that grants are won to do this work. Companies are founded to push the work into product production and so on. I think that there's a lot of bad biology, and I'm not saying that this paper is bad biology. It's interesting biology, but the inferences that are made about what it could be used for are often based on bad biological extrapolations, and so there's that. There's a tremendous push to commercialize things.
Yes, and that's really the carrot, isn't it? It's when you're talking about how grants are won. Grants are won on big promises. The bigger the promise, the more likely you're going to get the grant. And the big promise is we're going to solve all these big diseases. I've heard people talk about using human, non-human chimeras for Alzheimer's and Parkinson's and cancer, and as you say, organ replacement and that sort of thing. And there's a wow factor there. And yet, you first challenged this in 1997. Here we are in 2020, and it's creeping along, I suppose, would be
and I think
describe it.
And I think the frontier ahead of us that they really want to implement is what's called human germline modification, where they go in with genes using maybe the CRISPR technique or something like that, and modify embryos, possibly modify the sperm or egg cells before fertilization, or modify the embryos after they're established, so that they develop into something different from what they would have had they not been genetically engineered. And here's where bad biology comes in, because genes are not independently acting modules. And even if you have a gene that you think you know the function of, if you put it into a new context, its operation can be very different from what you expect. And there's no such thing as a kind of a pure genetic background. So every time you do this, you're doing an unprecedented experiment, and you're just hoping for the best. Sometimes this is going to happen, and it's going to turn out to be totally different from what is expected. So then, what do you do with that embryo or that child or that adult who at 30 turns out to have some awful disease because of some unexpected manipulation?
But isn't it also the case that we've gone down a route of thinking that genes are the be all and end all, genetic determinism, that our genes determine everything about us, and therefore all of our research is being poured into finding out the genetic origins of things, when in fact, at the side of this research is plenty of new data coming in suggesting that that's not the case.
Right. So genes don't determine everything, and in fact, gene differences don't determine everything. Sometimes the same gene in two different people gets modified differently. So it's exactly the same gene, but because of other components of the individual's body, the genes get used differently. So the genes are not a program, and they're not a blueprint, They're none of that. And nobody denies that you can change things by changing the genes. I mean, that's very clear. But in what way do you change them? And if something looks good for the first 2 years, will it stay good after that? Humans and other animals develop throughout their whole lifetime, and genes are used differently at different stages of life. So we're really very far from any ability to engineer complex organisms. And I don't think we'll ever really be there. I think engineering might not be an appropriate concept for organisms.
That's interesting. It occurs to me again, listening to you, that one of the things that the pandemic has highlighted for many of us is that this kind of extreme science is happening everywhere all the time with very little oversight and very little public awareness, and yet the consequences of it are potentially very far-reaching. So it's great to know that there are science watchers like you on the case, but isn't what we really need a kind of rational and consistent regulation throughout the world? We saw with the gain of function research, it's no good one country banning it and another country embracing it, particularly when there are still so many open questions about it. So where are we with the legalities and the regulations around this? Have things changed since '97?
Well, I think things have changed. You see that dictators can rise and just throw away regulations, even if they're there. So it's very unfortunate. It may not be regulation, but somehow just a kind of a public awareness, widespread education and rejection of certain routes of research. So it may be that you can't stop somebody from doing something in their garage, but if there were taboos against doing certain things, people just wouldn't accept the products. And people wouldn't, if people understood this well enough, they just wouldn't present themselves for genetic engineering or for the genetic engineering of their offspring. Or there's a widespread international rejection of genetically engineered crops, even in places where it's not against the law. A lot of culturally, a lot of people just have misgivings about it for various reasons. And so it's just that kind of awareness that is going to make people reject certain ways of doing things.
And so maybe one of the answers is to bring more people into the conversation. I mean, the general public tends to be excluded from these conversations, and
Exactly.
If them would find it technical and a little overwhelming. And yet, I don't think people are short of opinions on it. So is there any possibility one of these days of having a kind of citizen panel that oversees or looks at or contributes to some of the regulatory issues around this?
Well, that would be very good. I mean, this should be talked about very widely. And the kinds of things that are at issue are not the technicalities. Sometimes people with interests in doing it will attempt to razzle dazzle the public with one way or another. Like instead of saying how dangerous something is, they'll say how non-dangerous something is, and that's the tendency when things are commercialized and when grants are at stake. So, for example, with CRISPR, CRISPR is very much more accurate than a lot of other genetic engineering techniques, but it's not infallible. And when it was announced that how good it was, immediately people were saying, and we can genetically engineer people with it. And then papers started appearing about how fallible it was, how fraught with errors and inaccuracies and so on. So then every paper that made some headway in this regard would say, and now it's ready to do it with humans. But it's not, and I don't think it ever will be. And even if it's made perfectly technical, and this is something the public could understand very well, even if it was made absolutely accurate, a technique like CRISPR, the complexity of the organism that is being supposedly engineered is too great to have a predictable outcome or a completely predictable outcome. So there are intrinsic uncertainties, not simply uncertainties in the technique, but intrinsic uncertainties in the whole enterprise.
Yeah. Well, we're about coming to the end of our time, Stuart. I guess we're just going to have to keep watching and relying on good people like yourself to help us make sense of the issues. But thank you so much for coming on and talking to us about this today. I guess watch this space.
Yes, thank you.
Thank you.